In this lesson, we'll be talking about selective and differential media. So we're going to be focusing on mannitol salt agar, McConkey agar, eosin methylene blue agar, and blood agar. The first type of media we'll talk about is the mannitol salt agar, or the MSA media. So this is a selective and differential media, meaning it only allows certain types of bacteria to grow, but of the type that can grow, they will typically grow different colors based on how they interact with the contents of the media. The MSA plate specifically is used to isolate and identify members of the Staphylococcus species. Typically, only members of Staphylococcus are able to grow well on this plate. The plate has a high salt concentration, hence the name mannitol salt agar, and most bacteria are not able to thrive in the presence of salt. So most bacteria, like E. coli, if you put them in this plate, they would not be able to grow. It would create a hypertonic environment, and the bacterial cells will shrivel and die. The exception to that rule, though, are Staphylococcus bacteria. Staphylococcus bacteria are what we call halophilic, meaning they're able to survive in high salt concentrations. So if you put any members of the Staphylococcus genus in this plate, they will typically thrive because of their halophilic nature and their ability to survive in high salt concentrations. So this plate is specifically used if your intent is to isolate and grow members of the Staphylococcus species. Now there are different species of Staphylococcus within the Staphylococcus genus. So for example, there's Staphylococcus aureus, which is typically a more pathogenic species of Staphylococcus, causing infectious disease throughout the body. And then there's something like Staphylococcus epidermidis, which is just a typically a healthy skin biota. So we do need to talk about how we differentiate from what would be considered like pathogenic Staphylococcus, like Staph aureus, and kind of normal biota Staphylococcus, like Staphylococcus epidermidis. So all different types of Staphylococcus are going to grow on the mannitol salt agar, again, because they're halophilic and they're able to survive in the high salt concentration. However, they will grow different colors depending on if they utilize the mannitol as a source of fermentation. So if you're working with something like Staphylococcus epidermidis, which is considered a normal biota, it will grow in this plate because of the high salt concentration. However, S. epidermidis does not ferment mannitol. Since it does not ferment mannitol, it typically is going to grow this kind of light pink color on the surface of the mannitol salt agar. Then we look at something like Staphylococcus aureus. Staphylococcus aureus, it is able to ferment mannitol. So when you put it in here, it's going to grow because it's able to grow within the high salt concentration. And then it also ferments the mannitol as an energy source. When it ferments mannitol, acids are released, which cause the pH in the media to go down. And when the pH in this media goes down, the pH indicator called phenol red will cause the plate to shift from this pink color to a bright yellow color. So when we see yellow growth, that's an indication that mannitol has been fermented. So again, with something like Staphylococcus epidermidis, it's going to grow because of the high salt concentration. However, it does not ferment mannitol, which means acids do not get released, which means the pH does not go down. And because the pH doesn't go down, the color shift doesn't happen and the growth just remains pink. But in cases where mannitol is fermented, for example with Staph aureus, those acids would be released, the pH would go down, and that pH indicator would shift to yellow. The next media we'll talk about is the MAC agar, or the McConkey agar. This is also a selective and differential plate that is used to isolate members of the Enterobacteriaceae family, which is just fancy terminology for bacteria that are commonly found within the gastrointestinal system. So the McConkey agar, it is selective and differential. It's selective because it only allows certain types of bacteria to grow, but also differential because different types of bacteria will grow different colors. So we're going to first talk about why it's selective. This plate is selective because it only allows gram-negative bacteria to grow. Gram-positive bacteria are inhibited from growing on this plate, which makes sense because the purpose of this plate is typically to grow gut bacteria, which are predominantly gram-negative bacteria. Gram-positive bacteria are more common for like the skin and upper respiratory system, but gram-negatives are more commonly found in the gastrointestinal system. So when you put a gram-negative bacteria in here, it's going to thrive and grow really well. However, gram-positive bacteria, which would have been put over here, are inhibited from growing because of two things, bile salts and crystal violet. So bile salts and crystal violet embedded within this media are going to prevent gram-positive bacteria from growing. So if you do inoculate this plate with a gram-positive bacterium, it wouldn't actually be able to grow. So this plate is in part selective because it inhibits the growth of gram-positive bacteria using bile salts and crystal violet. So we know only gram-negative bacteria are going to be able to grow on this plate. However, different types of gram-negative bacteria will grow different colors depending on how they utilize the contents of the media. So MacConkey agar also has the sugar lactose. So some gram-negative bacteria will ferment the lactose as an energy source, and then obviously some won't. So similar to the mannitol salt agar, there's also a pH indicator embedded in this plate, and when the pH goes down within the plate will shift colors, which typically indicates that lactose has been fermented. So in this plate, if you put a gram-negative bacterium in here, and it does ferment the lactose, acids get released, the pH goes down, and this pH indicator called neutral red will actually produce bright pink growth. So in a MacConkey agar, bright pink growth is indicative of a gram-negative bacterium that is able to ferment lactose. Over here, what's happening is a gram-negative bacteria was inoculated, so it was able to grow. However, it does not ferment lactose. Since it does not ferment lactose, acids are not released, the pH does not change, and the growth is simply kind of this colorless growth on top of the surface of the media. So only gram-negative bacteria will grow. However, if they grow pink, that's a sign that lactose was fermented. If they grow this kind of colorless appearance, this translucent appearance, then that's typically a sign that the gram-negative bacteria does not ferment lactose. The next plate is the eosin methylene blue, or EMB plate. This plate is also selective and differential. Selective because only certain types of bacteria can grow. Differential because they grow different colors based on utilization of the sugar contents within the media. This plate, very similar to the last plate, is used to grow bacteria commonly found in the gastrointestinal system. Specifically, this plate is used to isolate fecal coliforms, or bacteria commonly found in feces. So this plate is in part selective, so it only allows some certain types of bacteria to grow. Specifically, it only allows gram-negative bacteria to grow, just like the last one. Again, we're looking for fecal coliforms, so that's going to be predominantly gram-negative bacteria. So we want to inhibit those gram-positive bacteria from growing. We can see here a gram-positive bacteria was inoculated here and it did not grow. The reason why gram-positive bacteria cannot grow on an EMB plate is because of two things, eosin and methylene blue. Both of these work together to inhibit gram-positive growth. So as soon as Staph aureus was inoculated on this plate, it essentially just shriveled and died because of the inhibition by eosin and methylene blue. So we know only gram-negative bacteria can grow on this plate, which we have 3 different types of gram-negative bacteria here that we're able to grow on the plate. However, they will grow different colors based on how they interact with the contents of the media. So if we look at this first one here, it grew kind of this metallic green color. Typically, gram-negative bacteria that grow metallic green or even like a dark purple to black on this plate, that's a sign that rapid lactose fermentation has occurred. So they've very quickly and efficiently fermented the lactose. So that's typically consistent if you see this kind of metallic green or even black to kind of purple coloration. And we can see that here in quadrant 1. In quadrant 3 down here, we see this kind of pink growth. So this is a gram-negative bacteria that weakly ferment. It's a weak lactose fermenter. So it does not ferment lactose rapidly, although it does ferment lactose. So typically with this type of plate, if the growth was a pink color, that's an indication of weak lactose fermentation. And then there are some gram-negative bacteria that are going to grow on this plate. They just don't ferment lactose. If they're able to grow on this plate, but they do not ferment lactose, then they're going to kind of grow this translucent color. So again, if it's a gram-positive bacteria, it's not going to grow because of eosin and methylene blue. If it's a gram-negative that rapidly ferments lactose, it'll grow metallic green, black, or dark purple. If it's a gram-negative bacteria that weakly ferments lactose, then it's going to grow pink. And if it's a gram-negative bacteria that does not ferment lactose, it's going to grow a translucent or colorless color on top of the agar. The last plate we'll talk about is the blood agar. The blood agar is used to see if a bacterium has any type of hemolytic toxins that can destroy red blood cells. There are several different species of bacteria that have what we call hemolysins. The most commonly known one is Streptococcus pyogenes. This is the bacteria that causes strep throat, but it has what we call a beta hemolytic activity, which means it can completely destroy red blood cells. So when you put it in blood agar, the bacteria here destroy the red blood cells all around, and you kind of get this clear halo that forms around the bacteria where the red has been destroyed by the hemolytic activity. So there are 3 different hemolytic types. There's beta, alpha, and gamma. So beta hemolytic activity means there was complete destruction of the red blood cells. So when you inoculated the bacteria into the plate, it released the beta hemolytic toxins and completely destroyed these red blood cells. So you get this clear halo that forms around where you inoculated the bacteria because of the complete destruction. There's also alpha hemolytic activity, which we see here. Now this is partial destruction of red blood cells. This is common for something like E. coli. So they only partially destroy the red blood cells, which results in this kind of green discoloration of the agar. And then there's gamma hemolytic activity, which is really no hemolytic activity. In gamma hemolytic activity, the bacteria just grows on the surface of the blood agar, but it does not release any toxins to damage the red blood cells. So we can see the bacteria growing, but then we see this rich red coloration all around, and that's because the red blood cells have not been destroyed at all. So in beta hemolytic activity, there's complete destruction of red blood cells. In alpha, there's only partial destruction of red blood cells, and then in gamma, there is no destruction of red blood cells.